Hello, everyone. Good afternoon. Thank you for being here. We will give you today hopefully simple and short update on the research we are doing, mainly in Finland, and then my colleague Anja from Zurich. And as you can see from the title, we will start by stressing the fact that SARS-CoV-2 does not infect human neuron efficiently, and we take this as a good news, as compared to other viruses that we call more specifically neurotropic. SARS-CoV-2 does not seem to belong, strictly speaking, to that category. My laboratory is mainly in Finland, in Helsinki, and I also have, as Lena suggested, a laboratory in Australia, the opposite poles of Earth. Today, we will start by considering the type of experiments that we do in our consortium, and I will talk about mostly experiment done on cells in a laboratory, whereas my colleague Anja will move into more in vivo environments, so in animal work. So what we know so far, does SARS-CoV-2 infect our brain? Well, so far we know that SARS-CoV-2 does not seem to at least spread efficiently in the human brain. I think this is now well established. If you compare, as you can see in my schematic here, what this virus does in the respiratory tissues, for example, in the lung or in the upper respiratory tissues, one cell gets infected, virus replicates very efficiently and spreads to the next, and each cell produces large amount of viruses that can either infect the neighboring tissues or go deeper into the respiratory tract and then come out of the first infected person and spread to the next via aerosol. Okay, whereas in the brain tissue of people who have diseased after COVID-19 and have been undergone autopsy, we hardly saw any virus in actual brain tissues. As I will show you in a moment, there are some reports of trace amount of virus, but not really something that would speak for a real efficient infection of the brain. And so let's summarize before we dive into what type of experiment we do and the results, and what is essentially long COVID and the main neurological symptoms that led us to ask the question whether there is a virus that could potentially go into the brain. And these are the experiment we want to do. So long COVID or post-acute COVID-19 syndrome essentially refers to the long-term consequences of the SARS-CoV-2 infection. And what is very clear by now, but already a few years ago, that it is a condition that affects multiple systems in the body. So in a summary here, you have the bibliographic reference at the bottom right corner of the slide if you're interested in reading more. Essentially, in this schematic, I wanted to emphasize that the symptoms that are classified under the big umbrella of long COVID refer to conditions that affect multiple organs. And the one that we are most interested in are the neurological system, mental health, and of course respiratory, but also other less defined symptoms like general fatigue that affects basically a diverse amount of people who are classified as long COVID patients. And this is actually the common theme. Basically, how many people have long COVID and what kind of symptoms they have. It changes a lot between one country and another, between what kind of person and medical history you have, and therefore it is very difficult to define and study a disease of this type. The WHO, so the World Health Organization, in 2021 gave these three guidances. The symptoms that have usually occur after 3 months from the onset of probably and confirmed SARS-CoV-2 infection. So if symptoms sort of persist months after you were positive, that's the first condition. Then they have to last for at least 2 months, and they cannot be explained by alternative diagnosis. And this is the difficult part, because it is not always easy to know the entire medical history of a person. So you can imagine that it's not so easy to even define what this disease is, but this is why we put together such a team, is to see whether we can find quantitative ways, scientific ways to define this disease so that we can better define it, study it, and therefore treat it. It is not easy to estimate the numbers that change with time and country in terms of how many people have it. For example, a recent study from Norway suggests a little bit more 6% of the COVID-19 infected people develop long COVID, but other studies, including our statistics, suggest that it might be less than 1%. So you see here, it depends on how you define it and what the threshold of definitions are from one country to another. These numbers can change. But clearly, I think, at least I personally believe, and many of my colleagues believe, that it is nothing near the numbers that were initially given, like 30% or something like this. It is much, much lower. And good news is that vaccination with any COVID-19 vaccine seems to reduce significantly the risk of developing long COVID. So the vaccine is not only good to prevent serious disease and death, but also to reduce the risk of developing long COVID. So what could be the cause of these neurological symptoms? We can define two main possibilities. One is that there is a general systemic reaction of your body, let's say a big immune response or something that goes throughout the body. For example, the infection of the respiratory tract and other tissues, long-lasting immune responses, multi-organ dysfunctions, including the brain. So something like a global reaction of the entire body. That is one possibility. The other one is that maybe there is a very limited brain infection. Let's say some area of the brain could be infected. Possible neuron damage could occur. There could be a brain immune response which could cause brain dysfunction. So these are the two questions that we asked at the beginning of the project. And my team and the team of my colleague Anya were basically tasked to study which of these two could be directly linked to an infection of virus in the brain. So essentially the second option. So what do we know so far from people? Well, people that have died in an hospital settings and gave the permission to do autopsies allowed some studies to reveal that there are some traces of viruses that will be found in some different organs, sometimes including the brain. However, not all the studies confirm this possibility, this fact. So in some studies, they did not find any evidence for actual brain infection. So it is not conclusively proven that there even is any trace of virus in the brain of any of the COVID-19 patients. So this is something that is important to notice, that if there would be a very strong infection of the brain, we would have found it very, very easily with the modern techniques. But this is not the case. So that's a starting point. And therefore, we can exclude a strong infection of the brain. But of course, it is our task to study even if the very limited infection might cause problems. So can the virus infect any neurons? And if it does, what happened to the neurons? This is the task that I will talk about today. Before we dive into the SARS-CoV-2 and what it does to a human neuron, we have to remember that there are many viruses that are known to infect the brain quite efficiently. Rabies, it's the most famous one. Poliovirus can cause paralysis, herpes simplex, Epstein-Barr, Zika, and so on and so forth. They are not very common, but they are not even uncommon. It can happen. And so, in general, how does the virus reach the brain? Well, there are basically 4 main routes that have been proposed as the mechanism. One is that from the blood, viruses that are circulating inside our brain vessels, which are then protected by the blood-brain barrier, if this barrier is leaky, if there is a structural problem, then the viruses can leak into our brain. That's one possibility. The second one is that there is a portion of our brain that produces the liquid that goes in between the cranium and the the brain, which is the cerebrospinal fluid, and these cells that are in the choroid plexus could allow a virus to spread and access the brain. Then that could be cells that are immune cells that through the vasculature route could reach the brain. And then there's the nasal route that my colleague Anja will tell you about more in detail. So 4 possibilities, and essentially the question from my laboratory was, if, hypothetically speaking, this virus would enter the brain, can it even infect neurons? If it does, what does it do? And can we find treatments to prevent that from happening in case it would? So in addition to the to the routes that I described before, there is also a possibility that very long neuronal connection that from the spinal cord through each of the organs could lead a virus to go backward up from one neuron to another. And this route is, for example, used by rabies virus, polioviruses. They can use neurons to go back to our brain. So in my studies, we also studied if a virus can efficiently spread from one neuron to neuron to see if this possibility could be taken into account. And so the first take-home message from our study is a good news again. SARS-CoV-2 does not spread efficiently in the human brain. And this is now very clear from all the autopsies. And limited infection could potentially cause symptoms, and this is what we want to study. To do that, we have to ask specific questions. Each of them can be then addressed by specific experiments. For example, if it is true that this virus would infect the neurons, how efficiently does SARS-CoV-2 infect human neurons? How does it get into the neurons? Can it infect all the brain cells, for example, astrocytes, microglia cells, and so on? What happens to an infected neuron after it is infected? What happens to the infected brain? This is actually a question that Anja will tell you a bit more. And if it happens, how can we stop it? Okay, so these are the basis of the studies that we have set. And how do we study an infection in the brain? Well, we need to have model systems. So in the laboratory, we can create out of stem cells neurons and astrocytes and have basically laboratory systems in which these cells grow in two-dimensional cultures. And we can see what the neurons are thanks to fluorescent microscopy. But then we can create in a model system that are a little bit more complicated, as we call them mini brain or organoids. And this is work that I do in my laboratory in Australia. Essentially, I will tell you in a moment about this, but this little sphere that is about 2 millimeters long contains essentially the main cells of the brain. So it is a model system to study how things happen in 3 dimensions without the use of any animal. And they are human cells. The third is the one that you absolutely need if you want to understand what happens in vivo. And this uses, of course, preclinical animal models that we try to use as minimum as possible and under absolute strict rules of ethical considerations and regulations. And this is the work that Anja Kipar will talk to you about after me. So the human mini brain has a model system. I told you before that the brain is made of neurons, astrocytes, and immune cells of the brain, the microglia. But then there's this blue liquid that you see here. In reality, it's transparent. Here I color it in blue. And there is an epithelium called the choroid plexus in 2 parts of the brain that produces this liquid. And it is a possibility that these cells could be the ones that mediate the infection of the brain. So we created in a laboratory setting, starting from stem cells, mini 3D spherical structures that contain neurons inside, in the dark part. You see the neurons in orange. You can see if the neuron is infected because we use techniques that allow the infected cell to become fluorescent. So you can see it here in green. This is an infected neuron. And then in these little sacs is the choroid plexus cells that produce really liquid inside. It's the cerebrospinal fluid. So we can study at least the component of the brain in a relatively simple three-dimensional system. What have we learned from these studies? We know that the virus infects the neurons very poorly, but in this schematic where you can see in white the neuronal cell, in blue the astrocytes that support the neurons, and then this one here is an immune cell of the brain, a macrophage cell called microglia. So what we've learned that the virus enters the neuron, goes into the stomach of the neuron, and then inside this compartment there is a big change in the structure of the virus that allows the virus to inject the genome into the cell. And once this genome is in the cell is infected. We've also discovered two inhibitors, so two medicines that block either this step, so the entry and the injection of the genome, or the release of new virions. By combining these two, we have a very powerful tool to stop infection. So if there would be a viral strain in the future or a new coronavirus that would infect neurons more efficiently, at least we have a good starting point, these two new drugs that we have characterized. Once the viruses are produced and come out, the astrocytes and the neuron produce certain chemicals that alarm the sentinel cells, the microglia, that become activated. So we can prove in the laboratory that even if neurons are infected very poorly and the virus doesn't spread well, the presence of the virus is sensed by the surveillance system of the brain, which becomes activated. You can see in these two graphs on the right side that when we then add to our cells an increasing dose of the two drugs that I've mentioned before, the CA8074 and the IMP1088, the infection levels that you can see here on the vertical axis, they go very quickly down with relatively low doses, actually. In this one, you need a bit more. The other one is very potent. Combining the two, you basically block infection completely. And they are well tolerated by the neurons. So the results so far. Infection of neurons is very inefficient. It's less than 0.2, 0.1%. And to give you a comparison, if I would have, when we use a real neurotropic virus like Zika virus, you infect more than 80% of the cells within 2 days, whereas here you have to wait until 5 days to see a couple of cells infected. So clearly, this virus has not evolved to or has not learned the ways to infect neurons very efficiently. And that's obviously a good news. The receptor to enter the neuron, the main one, remains the same receptor, ACE2, that is used in the lungs, but neurons express very low level of this, so there might be another receptor that allows this little bit of infection to happen, or at least that helps. Once the virus attaches to the outside of a neuron, it needs to be eaten up by the cell in order to infect them. Sorry. We know that we have now two inhibitors that block infection, and once the neurons are infected, they have a relatively mild immune response. So very, very small reaction to the virus. Now, in order to validate these initial results that we have obtained in this beautiful, simple model system, we have to see what happens in vivo once we infect animal models and see what we can learn from that. Do we get the same results? You will see it in a moment from the next talk that is from my colleague Anja Kipar. And meanwhile, I will acknowledge some of the people who are involved in this work, mostly from my laboratory here on top in yellow. Then many people in Helsinki from different nationalities. These are very international projects, which is extremely motivating. And then collaborators in Australia, Anja Kipar, of course, that you will listen in a moment, and all the rest of the collaborators around the world. And of course, our funding agencies that provide us the money to complete the work, including, of course, the European Union Horizon program, which is the one that is funding this project. Okay, thank you very much for your attention. And if there are questions, I will be very happy to take them.